I have a loaded question here. What's the most common source of misleading GPCR data, and how do you prevent it? Yeah, well, my old boss used to say you need three things in experiments: controls, controls, and more controls. GPCRs are sensitive reactive proteins. They react to a lot of things, but you have to first show that your effect is selective for the target. And so you check your cell effects and selectivity and so forth. Then realizing that if it's an agonist or antagonist as well, it's a partnership between your compound and the sensitivity of your tissue. So you will get different profiles in tissues of varying sensitivity. Now, if you don't check that, your compound will meet those different tissue sensitivities anyway when it goes in vivo, and you might get a surprise there. So check all you can about the efficacy of your compound, of affinity in systems of different sensitivities. People have been fooled by just sticking with a high expression system and then moving on from that. In this age of biased signaling, too, you can no longer assume that a single signaling pathway will be all you need to check, because your valuable effect might be on some other pathway. So it certainly is worthwhile checking to see if you have a biased agonist. Having said that, I'll tell you almost all agonists are, and it's a question of what your pathways are. The most common way to do this is something like G protein, cyclic AMP, for example, and beta-arrestin. These are the standard workhorses. You can then ask, well, what if I don't want to have an effect on either of those? Doesn't matter. Those are your sensors for your agonist making a unique active state on formation. That's what you are after. So that'll tell you this agonist makes a different active state than the normal natural hormone or transmitter. That is going to play out, or it could anyway, play out with differential effects in vivo. So that will mark that compound for special treatment. So the question was misleading data, and I'm giving you a treatise on the cautious, correct way to look at a GPCR response. But failure to check those things can get you in trouble.
Absolutely. And if I can add one other thing, I think the assay that you're using as a readout plays also a very important role, especially depending on the stage at which your program is. Very early on, you might start off with molecules that are weak, so you might want a system that is very sensitive, that detects what you need. And to your point, Harry, controls. But sometimes the controls are, well, most of the time the controls, especially positive, are well characterized, well optimized. So you will always have a signal. When you run your control, you want to figure out how you set up that assay and what kind of readout you want to have in order to make sure that you capture what your molecules are doing and have a baseline, a reproducible set of data that allows you to then build upon that.
Right. Good. Yes.